exam that I caught when I was grading. Let me fix this real quick. Which one? Okay. I think this should be good. All right, so let's start. You guys stop me as you have questions, okay? But we're just gonna go through these multiple choice. And then any other questions y'all wanna go over. I'll go over the whole exam, but we have about an hour or less to play with. So number one, obviously, was just a fun question. So any answer I accepted there. In my opinion, the best answer is a sugar-free bag of heartbroken gummy bears. But number two, classify each amine labeled 1 and 2 in the structure of lidocaine, a local acting anesthetic, as primary, secondary, or tertiary. By the way, do y'all have y'all's emails? Did it come through? Can y'all see your exams? Okay. So number one, this amine right here is bonded to 1 carbon there and 1 carbon there. So that's secondary. This amine right here has 3 carbon bonds. I'm going to highlight them. 1, 2, 3. Okay, so that's tertiary. So the correct answer was B. Amine 1 is secondary, amine 2 is tertiary. Okay, some people, not everybody, but a few people missed number 3. In organic and biochemical reactions, water is often used to separate a molecule into 2 parts. So we're not saying water is a product here. It's just water is being used to separate. So that process has a specific name, hydrolysis. And we've seen that with alkenes. We've seen that with carbohydrates. Now today with peptide bonds, we can hydrolyze those. And it's a very common process. So that's why I put it on the exam, because we're going to continue to hear about it as we move forward into biochemistry. So questions on that? What tripped people up on this one? You may not want to say, but
I thought it was esterification because when I looked, like what I had on my note card, but then after the test, I realized it said the reverse was hydrolysis, and I felt dumb. So.
Oh. Well, at least you know what it is now.
Yeah.
All right, that was number 3. Which of the following is the correct formula for potassium stearate? So potassium is K plus stearate. So the correct answer you obviously see is A, but this should have been right on your resources page, which I could scroll down to. It's at the very bottom. Stearate is right here. So CH3, CH2, 16, and then COO. Now this is stearic acid. So when it becomes stearate is when it loses this hydrogen right here, and it becomes the anion form. I should write that next to the oxygen, okay? And then the positively charged potassium is what is attracted to that negative charge. So that's potassium stearate. So if you pick any of the forms that had the hydrogen still attached, that was wrong. That's stearic acid. It's stearate when it's the carboxylate anion form. Okay? Okay, I'm gonna scroll back up. Number 5, so the only way I messed with you guys was by changing this number right here or leaving the H on it. And that both of those are wrong. The breakdown of triacylglycerols with a strong base such as NaOH is a process known as saponification. Okay. We talked about that Friday and gave an example of it. There was a figure and a slide directly on it. We defined it. It was a fill in the blank on one of the slides. But I think most people got that. Okay, which of the following compounds is acidic? This was probably one of the more tricky multiple choice questions. So I want to look at these. This is an ester. None of the hydrogens here are acidic. Every hydrogen on this molecule is bonded to carbon. None of those are positively polarized hydrogens. So this one's not acidic. Number 2, we have it's an amide. But specifically, it's an amide with a plus charge on it. That nitrogen has 4 bonds. And so it's an ammonia type of compound. And we said any compound where the nitrogen is in an ammonium ion form is weakly acidic. So it's not a strong acid, but it is weakly acidic. So these hydrogens, one of these 2 right here, could be donated in an acidic as an acid would. Questions on that? Let me see if I can find the that was specifically in where we talked about amines. Acidity amines, nope, it was just after that. Amine salts. Wait a minute. Right in here. Here's where we talked about ammonium ions, but oh, right here. These are reversible. So the reason was that the ammonium ions can react as acids in the presence of a base. So that is where we mentioned that particular aspect of ammonium ions being acidic. Okay, this is a carboxylic acid, so you for sure should have picked number 3 as acidic. Okay, we know this hydrogen right here is acidic. And then this one is just an alcohol, specifically a phenol, but alcohols, this one's not acidic. Okay, so D was the correct answer because 2 and 3 are both acidic. All right, which of the following carboxylic acids would be most soluble in water? So this one has the longest hydrocarbon portion. There are 6 CH2 groups, so that's a long hydrocarbon tail on that carboxylic acid. This one only has 3 CH2 groups in that terminal CH3 group. This one only has a CH3 and a CH2 group. So this is the smallest one. This one's the most hydrophobic or hydrophilic. This one's the most polar. So the correct answer here was just C, that number 3 would be the most soluble in water because the hydrocarbon portion of the molecule is the smallest there. D, I think most everybody got D. You just count the carbons, 1, 2, 3, 4, 5. So this is a pentose. And specifically this right here is a ketone group, not an aldehyde. So it's a ketopentose. So D was the correct answer. Which of the following monosaccharides, number 9, is found in both plants and milk, metabolize the glucose? You could have written this on your card maybe, but this is galactose. Galactose is found in milk, found in plants as well, metabolized to glucose. And then 10, the reduction of a monosaccharide with a reducing agent, a sugar alcohol. So that's where we made a big deal about like sorbitol, and we said sugar alcohols, if you consume a lot of them, they can not do great things for your body. But sugar alcohols, so when you have this aldehyde group on, or sorry, aldehyde group on monosaccharides, you can reduce this. You can reduce this to an alcohol. H there too. So that's sugar alcohol. So the correct answer there was sugar alcohol. Okay, any questions on the multiple choice? And why the right answer was the right answer? So this is reduction. Why is this reduction? Because you have 2 carbon-oxygen bonds here, and you only have 1 there. So we said oxidation is increasing the number of carbon-oxygen bonds in a molecule. Reduction is the opposite. It's decreasing the number of carbon-oxygen bonds in a molecule. Or increasing the number of carbon-hydrogen bonds. Okay, 11. This was all about amines. This whole problem was about amines. So an amine with 3 bonds in a lone pair is a quaternary ammonium ion. That is false, okay? So that looks like 3 bonds in a lone pair. I don't even care if these are to carbon and hydrogen. This is not quaternary. Quaternary, it's got to have 4 bonds. And ammonium indicates it's got some kind of plus charge. But when nitrogen has 3 bonds in a lone pair, it's neutral. Okay, so that one was false. Amines have higher boiling points than alkanes, true, and lower boiling points than alcohols, true. Alcohols, the hydrogen bonding there is stronger, so it takes a lot more heat or energy. Maybe not a lot more, but it does take more heat or energy to boil those, break alcohols apart than it does amines, even though amines can also hydrogen bond. Quaternary amines do not act as acids or bases. That is true. Okay, so quaternary amine is an amine with 4 carbon bonds, or it's a nitrogen with 4 carbon bonds. So there are no hydrogens on this nitrogen. There's no lone pairs on this nitrogen. We said the lack of a lone pair and the lack of a bonded hydrogen means this type of amine is not acidic or basic. I thought this one right here was the trickiest one, but it was on a slide. It was straight from your lecture notes. In the body at physiological pH, amines are neutral. That's false. They're in their ammonium ion form. That's why, I mean, look at all those amino acids we just talked about. Here's the alpha carbon at physiological pH on the table. If you look at it, all these amines are NH3+. They're all ammonium ions. And we said this right here. At physiological pH, amine compounds exist as ammonium ions. Here's where we said that quaternary amines are not acidic or basic. Okay, neither acidic or basic. Lastly, an NH2 group attached as a substituent to a molecule is known as an amino group. That is true, okay? An NH2 bonded to some kind of molecule, that is an amino group. All right. Number 12. So here, let me explain something first. I ask you guys to describe the biological significance, okay? So what do I mean when I say the significance? Why is something important? You need to tell me the purpose of it. And we literally, the very first slide of our lipid lecture on last Friday, we walked through and we wrote in all the biological significance of these types of lipids. And then later, like when I got to steroids and things like that, we expounded on those as we went. So if you did not tell me the significance, if you just gave me examples like cholesterol and sex hormones, if you just said steroids, cholesterol and sex hormones, I counted off points because I want to know, what are they useful for? I don't care if you can list 20 examples. I want you to know why they're important in the body. Okay. So I didn't need a ton. I said, you know, one sentence should be adequate. You didn't have to write forever, but you at least need to mention why they're important. We gave several examples for steroids. We said cholesterol adds rigidity to the cell membrane bilayer. It's really important because it helps hold the bilayer together. We said sex hormones are responsible for the development of secondary female and male sex traits. We said bile acids. Nobody really mentioned this, but they act as emulsifying agents for digestion of fat. Somebody actually may have mentioned that on there. I can't remember. So there's 3 different significant examples of the functionality of steroids. Glycerophospholipids, I think most everybody got this really great. We said that they are abundant in the membrane bilayer, and they help regulate the flow of molecules into and out of the cell. Okay, so they help maintain gradients. I'm actually gonna not underline all this because I want to save this for later. Okay, so right here, regulate the flow of molecules into and out of the cell. They help maintain gradients across the membrane. All right, so eicosanoids, we said these are special localized cell messengers. Example we gave was prostaglandins. So they respond to inflammatory response when you get a cut or when you're injured. Blood clotting, they're involved in the blood clotting process as well. I don't know if I mentioned that or not, but I know I gave you the example of inflammatory response and told you that they were cell messengers. So I didn't need a lot there. Triacylglycerols, these are really, really important lipids. We're going to have a whole chapter on this when we get to metabolism of fats. But triacylglycerols are the lipids that the body uses for long-term energy storage. And metabolism of the cannot talk. Metabolism of triacylglycerols yields a lot of energy. Man, I've been hitting my word count by like 9:30 every single day when I teach summer course. So, any questions on what I wanted on this problem? Or if you got marked off a few points here or there, why you got marked off? And I want you to know, I am open to dialogue. If you feel your response was just, talk to me about it. Let's talk about it. Shoot me an email. I am happy to look over any part of your exam. Make sure I added things up correctly. I did this pretty quickly, so make sure that double check what I marked off and that it adds up to the right numbers. Always double check me. I'll be the first to tell you I make mistakes. Okay, number 13. This was a challenging problem for a lot of people on the exam. I don't know if anybody aced this question. So let's look at this. This is oh Lord. Okay, this is an ammonium. See the plus charge on the nitrogen? This is an ammonium compound. So we got bromide as the anion. So it'll be something, something ammonium bromide, okay? We said when nitrogens have a plus charge, instead of naming them as amines, we name it as we would an amine, but we change the ending to ammonium. So we are going to name this amine according to the substituent with the most carbons. And that is this one right here. That's the longest one. So we got 1, 2, 3, 4 straight chain, not branched. This is butyl. So it'll be something, something butyl ammonium bromide. And then right here, we got 2 ethyl groups. So this is NN diethyl. So some people forgot to put 2 N's, but I really didn't take off maybe half a point for that. NN diethyl butyl ammonium bromide. That's a monster of a name, I know, but that's what it is. Okay. This was, I guess, the hardest one. So this is an ester. So it should have an ATE ending. It's an ester because right here, you got the carbonyl carbon and then you got a carbon oxygen carbon bond. So the way you name an ester is you identify the group that the ester oxygen is bonded to. What is the substituent? What group is bonded to the ester oxygen? This is isopropyl. Okay, 3 carbons and you're bonded through the middle carbon right here, so that's isopropyl. And this ester, you got to consider, okay, what acid did my carboxylic acid portion come from? And if this was on your, did I put it? I think I put it on your table, your resources page. Right here. So here's the carboxylic acid portion, and when the only hydrogen, or when the only substituent attached on the other side of the carbonyl carbon is the hydrogen, that's formic acid. So as an ester, it becomes formate. Formate. So go back up. Should just be isopropyl formate. So that one gave people a lot of problems. Anybody maybe help me see where this problem was super challenging? Maybe it's just naming in general. Do y'all feel like naming in general for these chapters was just extra challenging?
I feel like,
go ahead, Claire.
I was gonna say, I feel like for the isopropyl formate, I think it was like the setup of it. Like I saw like the 3 carbons and I was like, oh, that's glycerol for whatever reason. I was thinking that in my head.
Oh.
but then I realized like, oh, that's not it. But I was like stumped on what it was. I couldn't like visualize. the
Gotcha.
formate.
Yeah, sometimes when you see a molecule a certain way, it's hard to see it any other way in your head. So I totally get that. So if you got any part of the word right, like if you gave me the AT, I'm on eraser. If you gave me the ATE ending, I gave you a point. If you said isopropyl, I gave you a point. Okay, it's just that many people just missed it all together. So
I struggled with naming for this chapter.
Cameron,
whole-
naming was really hard for this chapter. I agree with you on that it was hard. So this one, I tried to give you a few more specific instructions, like use the Greek letter and the common name, because we did practice that. So this was a carboxylic acid. This was actually right from your lecture slides. We named this one in lecture. I just grabbed the one. And here's the deal. I know what you're thinking. We literally have 100 lecture slides to look at. I'm not going to remember all the examples. I know. But there are times when I'll do this. So we named this one in lecture. It was a beta hydroxy alpha methyl. Then because there's 4 carbons, 1, 2, 3, 4, the common naming for this is down on your resources page. It was butyric acid. So if you spelled butyric wrong, I really didn't take off points. If you got some people got the beta and the alpha, you just put them in the wrong order. Like you said, alpha methyl, then beta hydroxy. But remember, you've got to do it in alphabetical order. So hydroxy first, then methyl. And butyric, so butanoic acid is the systematic IUPAC per name, and butyric was the common name, and that's down on your resources page. Okay. Questions on that? All right. A few questions left. Number 14. This one, 5 points each. So this one was a lot of points available here. And the first problem, you have an amine and an acid. So this technically is acting like your base. We said amines, they have lone pairs here. They act like bases. I think most everybody got this. This was your acid. So that lone pair is going to grab this hydrogen right there, and you draw the same molecules, just the hydrogen transferred over your conjugate base and conjugate acid, and that was that. Ammonium, an ammonium compound, and then your chloride anion. You didn't have to name it or anything. This, you have a carboxylic acid and you have an amine, so you're forming an amide. This one I saw kind of a few different things. So I just wanted you to, this is, you lose water, so water should have been a product. So right here's what happens. The OH right here, and I don't care, one of those hydrogens is gonna go. So that's where you get water. And then you just scoot the other two together and you bond it. So it should have been, draw the same molecule. Here's the carbonyl group bonded to the nitrogen, and then the hydrogen and the amine all the same. From that forms your amide. And I had written, Robin had asked me, what all reactions should we focus on? Are we going to need to focus on everything from our organic reactions trial? I said, no. I said, here's the ones you need to know. And I made a list. She said she took a picture of it and sent it to the group me. So there should have been a list. I wanted you to know these reactions. I put them right on this. I said, acid-base reactions with amines, amide formation, ester formation, hydrolysis of those. And then this one right here is ester formation. So this is esterification. Abby did something really clever on this one. I didn't count it wrong, actually. Abby, I hope it's okay if I share. I won't share. You can share if you want to.
I don't care. You can tell them.
Okay. So
I said, I guess so.
I didn't count her off because technically Abby said, oh, amines are bases and carboxylic acids are acids. So I'm going to treat this as an acid-base reaction. And she just drew an ammonium compound and a carboxylate anion. And I said, well, I didn't specify that you have heat or not. if I did write heat over this arrow, then you would have to tell me, oh, it's amide formation, but I didn't specify that. So I didn't count you wrong. So that was the one loophole on the exam, if you were looking for one. But I would wanted you to write this as an amide formation. Okay, this is a sterification. So all you're doing is, again, you lose water, this goes, and this hydrogen goes. You get water. And then you need to flip so we're gonna get a bond between that carbon and that oxygen. So that's what you see. This bond right here is that bond right there. And then this is your ester right here. Nothing else on the molecule has changed. This is not going to happen with any of these OH groups because these are not acidic. The only one that's acidic is the OH that is part of the carboxylic acid. So where you put this phenol group, or phenol group, I should say, onto the molecule, where you bound it was important. You couldn't just pick any of these OHs and rip one off and put it. You had to put it as part of the carboxylic acid so that you form an ester. So some people tried to attach it like through this OH, the aromatic ring and everything, but you can't do that. Okay. All right, this was a big problem. So I wrote out all three here. Nobody did number 3. So you all picked between 1 or 2. Let me talk about 1 for a second. If you did number 1, this was the formation of the disaccharide fructose alpha 1,4-galactose. So I said write the full reaction. So some of you only gave me the product. So when I say full reaction, I need reactants and products. If I only want one or the other, I'll just tell you write the products or write the reactants. So I hated that, because I feel like you could have written the reactants. I forget who I'm talking to, and it doesn't matter. But if you did not write reactants and products, you did miss points here. So you should have had fructose, and this was literally on your resources page. So you could have scrolled down and said, oh, there's fructose, and draw that. And then there's galactose, draw that separate. And now we're gonna put them together. The one thing that I was a stickler about here, because this was literally part of your carbohydrate lab, look at galactose. This is carbon 4. This OH is oriented up. Up. Fructose, this OH on C1, and these are the one that's important because it's an alpha 1,4 linkage, is pointing down. You have to maintain that orientation. The fructose has to be down, the galactose has to be up when you make the glycosidic bond. So what I saw a lot of, and it did cost points, was not this. I saw a lot of this. So if this is fructose, I'm not going to draw all the substituents on, but y'all would draw it like this. Okay, and there's galactose. So this is fructose and this is galactose. This is wrong. This is angled downwards, but on monosaccharides, you have to maintain the orientation of the OH groups. So I didn't take off a ton of points, but I mean, that was the main part of the problem. Like that was the main thing in your answer. So I might have taken off 2 points. And then if you had other like OH hydroxyl groups pointing the wrong way, I took off points. And literally all you had to do is copy that from your resources page down below and make sure you orient those the right way. But anyways, that was that one. Number 2, we had some people do number 2, hydrolysis of triacylglycerol containing 1 lauric and 2 palmitic fatty acids. So you literally just go grab those again from your resources page. You should have been able to draw the fatty acid portions correct. What you should have seen here, so this is the glycerol part. Looks like isopropyl alcohol, well, not alcohol, but an isopropyl group. It does. 3 carbons there, but and then this is your ester bond. So this is all formed together, and this is the lauric, and then these are the two palmitic. Okay, fatty acid chains, all attached to glycerol through their carboxylic acids. So when we hydrolyze this, what happens? We break these bonds right here. And water comes in, H and OH. The OH part goes to glycerol. So it's going to bind to these carbons. Each carbon on glycerol will get an OH. So you'll restore the glycerol molecule. This is glycerol restored. And then this hydrogen is what bonds to the carboxylate anion part to reform your carboxylic acid on the fatty acid chain. And so that's right there. These are the hydrogens from water. These are the OH groups from water, bonded onto the separate parts. So we just break it apart, and that was the hydrolysis. So if you messed that up a little bit, got most of it right, I gave you most of the points, but took off some. The hardest part with this is that the fatty acid chains are so long, it's hard to orient things properly the right way, but nobody did 3, so I'm not going to walk through 3, but that was hydrolysis of an amide under basic conditions. I didn't anticipate anybody would do number 3. I thought that was the hardest one, but I threw it on there just in case. Okay, number 16, describe the structural and functional differences between a micelle and a liposome. So the biggest part here was that I said structural and functional. I wanted you to tell me structurally, how are they different? And then functionally, what do they do? And why is what they do different? So you don't have to read my whole paragraph, but in short, you did not have to draw pictures. So I did not take up points if you did not draw pictures. Nowhere did I instruct you to draw pictures. Pictures always just help me. So I usually draw pictures for myself. But structurally, micelles have an internal nonpolar region because all the nonpolar fatty acid chains are oriented inwards, okay, because they're water fearing. And then the polar head groups, the carboxyl groups, are all pointed outwards. So you get this internal region that is completely nonpolar. Structurally, that's how it looks. Functionally, this is useful for trapping dirt and grease and nonpolar molecules, how soap works. That's the functional difference. Liposomes, most of you said this, have a bilayer. So they're composed of fatty acids as well, but these fatty acids arrange in a bilayer, which means you have this completely polar internal region. And that is useful for transport of polar aqueous hydrophilic molecules. They're happy, they're satisfied on the very, very inside of that, unlike they would be on a micelle. So a micelle, somebody could have told me transport of nonpolar things. A liposome, you could have said transport of polar things. Questions on that? If you only gave me the structural difference or you only gave me the functional difference, I did count off points. I wanted both. Last question. Describe the difference between active and passive transport. Be sure, some people miss this, and be sure to include examples of molecules that cross using active and passive transport. If you did not give me examples, I took off points. We mentioned a lot of examples in lecture, okay? And then discuss the significance of gradients and how they apply. So passive transport, most everybody aces. Passive transport does not require energy. Active transport does require energy. I'm really surprised at this point in the day that I can even formulate like a sentence that makes sense. Okay, active transport does require energy. Most everybody got that. And then a lot of people gave me great examples. You could have said small hydrophilic molecules use passive transport, small nonpolar molecules, carbon dioxide, oxygen, use passive transport. You could have said glucose molecules like glucose, amino acids use passive transport. Active transport is a lot of different types of ions, okay? Active transport, they're pumped. And the way the gradients play in is down here by my stars, okay? In passive transport, molecules flow down a gradient. The gradient is directing how the molecules flow. So the molecules flow from an area of high concentration to an area of low concentration, down a gradient. Active transport, the molecules are pumped against their gradient so that the gradient is maintained. You take molecules from an area of low concentration and you put them in an area of high concentration to maintain the gradient, and that takes energy because you're pushing them again. They don't want to go that way naturally. Naturally, they're just going to flow down the gradient. But when you apply energy, you can pump these molecules against their gradient to maintain the concentration differences. Okay, I'm tired of talking, and I know I'm surprised y'all have held on this long, so thank you for being attentive. The bonus was, so I curved the exam 7 points technically, because question number 1, I just gave you, well, okay, 4 points. Question 1 though, I just gave you 3 points there, and then 4 points here. So there was a small curve on this, but I think in a class this difficult at this pace, that's warranted, so. Okay, questions from you guys?